Taleb Barghouthi of Jordan. Dr. Barghouthi, for those of you who don't know, has been doing a human clinical trial for Verteporfin, which is a groundbreaking drug that has been shown in some studies from Stanford to regrow hair through wounds, through the ingreld 1 pathway. Uh, Dr. Barghouthi started his trial, I believe, back in June, right? It's going to be almost a year
Correct.
now.
Correct. So yeah, we're almost approaching 10 months, I think.
Almost, yeah, almost approaching 10 months. And I recall, so you have, if you could just explain sort of how your study has been, how it's been done. Like you have, I believe, 3 different dosages that you're testing, and you recently did a biopsy and everything. So if you could just kind of break down the different dosages um, for the public.
Yeah. So thank you for hosting us again, Melvin, and I hope the audience is with us here today. There has been a group of interested hair loss sufferers supporting us. So hello to everyone.
Yeah.
So we started essentially with an FUE study. Where we decided to categorize based on the animal studies a few of the promising doses that have been seen in animal studies. So we categorized the FUE sites into a 0.24 milligram, a 0.32, and a zero point 0.4 milligram dosage. And we did several 2 by 2 centimeter boxes where we punched out FUE grafts. In some of the control groups, we followed the same procedure that we usually do in a standard FUE. And in the trial or in the test boxes, we injected verteporfin in the corresponding dose that we were testing. And we followed up the results, initially at short intervals and then on a monthly basis, up to the point where in last month we did a biopsy just to get a better insight on the actual scarring mechanism that is in the skin itself.
Mm. So you did the biopsy, and what will the biopsy tell us for those for the audience members? Like
Yeah,
what is the biopsy gonna show us?
yeah. So we were, if you remember, throughout the follow-ups that we did, we used the dermoscope just to look at the scarring and to see any evidence of hair regrowth or potential hair regrowth, and to examine visually the type of scarring that is taking place or how the skin appearance and healing is showing to be. So at some point, we decided to even investigate a random area of the test and the control sites to
Mm.
see if there is any pathological or histological evidence of a difference between the test and the control areas. And that is done through a standard skin biopsy, which is 4 millimeter. It's bigger than a standard FUE punch size. So we took a 4 millimeter biopsy from the control site, one of the control sites, and a 4 millimeter biopsy from one of the test areas. Um,
Let's go ahead and
and
pull that up, Jerry. Oh, sorry, Dr. Bargathy. I just want to
yeah,
show the audience here. So this is right there where it says T is the test site. That's where you injected the verteporfin. C is the
yes,
controlled
correct.
site where you extracted grafts with no verteporfin injected.
Correct. You can actually see that the test site, the biopsy area is quite still clear. It's not as
Mm.
clear in the control area, but that's the scarring you see in the lower part of the test square
Yeah.
is a biopsy site. So because these pictures were just taken after the suture removal. Um,
Mm.
so yeah, so this is a 0.32 milligram area. I couldn't do biopsies on all 6 areas because it's a bit more invasive with biopsying. Um, each biopsy requires stitching or one stitch. So I just thought that, with the volunteer we'll just at least randomize one area, and we'll go with the. Luckily, it was the 0.32 because it's somewhere in between both doses. So
Yeah.
it could give a good sort of medium view of what's going on.
So, I mean, I think just from looking at the picture here, I think it's pretty visibly clear which side has more hair. Um, you know,
True, and this has been something that we sort of noticed throughout the regular visual follow-ups, let's say, and through the dermoscopy and the interval follow-ups. But I mean, it's a one patient study and it has its own limitations. So we just thought we needed to maybe add a bit more, objectivity to the study itself, and
absolutely.
see what's beyond the skin and what's underneath the skin. So the biopsy had two main sort of outcomes that we were looking at. One is to assess the scarring or the difference in scarring between both test and control areas, but at the same time to actually examine the type of hair follicles that are in such extracted areas, just to see if there's a difference in hair count, if there's
Mm.
what sort of hair is growing, if it can show that. And this is what we actually, I've shared the biopsy results on the thread. Um, and
On Hair Restoration Network, correct?
correct, yes.
Yeah.
We were able to show that there's, in terms of scarring, there was no peripheral follicular scarring or fibrosis in the test area with, as opposed to evident peripheral follicular fibrosis in the control area. And at the
Mm.
same time, the test area had minimal dermal fibrosis, while in the control area, the pathologist commented that there is obvious dermal fibrosis. So, in terms of scarring, this sort of comparison was, positive to say,
Mm.
and it did show that there are some differences in both sides. The choice of the biopsy site was random, so we were able to identify areas that have been punched. Um,
Mm.
so we just randomly selected 2 areas that looked like punch sites, FUE punch sites, and we just did the 4 millimeter biopsy. It's a bit bigger, of course, so it may
Mm.
have encompassed a couple of these punch sites with normal hair in between. But at the same time, if you see scarring on histology, you will comment on it. So especially that the pathologist did not really know what he's supposed to be looking at, apart from
Wow.
just FUE extraction sites. He was able to distinguish histologically a difference in the skin structure and the skin texture and the scarring itself. The other interesting finding was that the total number of hair follicles um, was double that in the test area compared to the control area. So we had 10
Wow.
hair follicles. I believe 3 were in anagen, 5 in catagen, and the rest in telogen. While in the control area, it was 5 in total. Um, so yeah, I mean that we can't base statistical significance on that, but there's clearly something to
But
explore.
the same number of extractions were done in the test area
Yes, yes,
versus the
we
controlled
did the
area.
same extractions in all 6 boxes.
So, I mean, I don't want to say, but I mean, you can extrapolate that if there's the double the amount of hairs in the test area versus the controlled area, that there was some regrowth. I would speculate there was some regrowth in the test area versus the controlled, right? I mean, that's what we can
Uh,
speculate. I mean,
so visually, when we were following things up, we could see better hair density in the test areas. So that was throughout. You remember the posts that were
yeah.
regularly updated. So visually, we could see that there's something different. But again, because of some limitations with the sites where the areas are, we couldn't track the same area every single time. This is something that we're gonna come to and fix in subsequent trials.
Mm.
But visually, we could see improved hair density, to a certain extent. But again, now in the same punch biopsy, and considering both areas are fairly similar in density, because these boxes are grouped together in the same sort of scalp area or region. So the hair density in each region was, almost similar, per square centimeter. So let's say we take a random biopsy, we'd expect to have a fairly similar number of hair follicles in that 4 millimeter punch. So to get twice that number is something that certainly requires further exploration.
Yeah, I mean, I feel like
And the
even if it's
interesting part,
randomized.
even and the interesting aspect is that almost 80% of these 10 follicles, for example, are anagen and catagen hairs. So, which means that, catagen phase is fairly short, which means it has been only recently into a catagen phase, and then it's going to progress to a telogen hair. So, it was only recently anagen, if you want to, conclude something. And so the majority have been, anagen hairs at some point.
And are they terminal hairs? Because that's another a question that a lot of people were saying. Like, are these hairs going to be like V-less hairs, like colorless small hairs, or are they
So
actual
the
terminal?
pathologist, I mean, he did not report on it, but he did say it's not anything different than a standard, donor area, terminal hairs. Because
Mm,
in the donor area, most of the hairs are terminal. So there was no difference in the sort of texture or the caliber of these hairs.
wow, this is. I mean, that's. I'm very excited. I mean, I don't know. We have a bunch of questions here in the chats. I don't know if there's uh let's start taking some questions here. Jerry.
All right. There's a question by Florin Productions.
Uh-huh.
He wants to know, can we pull up the strings here?
Can we pull up the string?
The strings.
I don't know what he means by that. The strings?
The strings. STAINS. This.
Pull up the
The
strains?
Sorry.
stains or stains.
Just yeah, I don't know what I'm maybe he can clarify next time because I don't know what he means by the strains.
Can we
What's
pull up
another
the
question?
stains?
Okay, some of
Oh,
these
you mean
hairs.
maybe he might be asking
Yes,
something in terms of
doctor.
pathology. Uh,
Oh, okay, okay. Maybe
like
so.
they do some staining when they test histological or pathological slides. I'm
Got
not
it.
sure if that's what he means.
Okay, maybe
Um,
that's what he means.
but to be honest, I'm not sure they actually stain them when they do that. I know there are certain pathologies they need to, but I'm not sure if um, in such to look at fibrosis or so they needed to stain anything, um, because they weren't looking for fungal or for a certain pathological um, uh, uh, you know conditions that would require this. So I can double check, of course.
Okay, perfect. And just so the audience knows, there's a website called verteporfin.org, which was created by users of the Hair Restoration Network to assist Dr. Barghouthi and his clinical trials. It's sort of like a crowdfunding website, which I'll also go ahead and link in the description when this stream is over. Let's go to the next question, Jerry.
Okay. Nimish Gar wants to know, to ask the doctor how we as a community can help him.
Okay, yeah, perfect. So how we can help is, like I said, go to verteporfin.org. And it's a crowdfunding situation where we can all contribute. I'm actually in the talks with Dr. Barghouthi as well. The Hair Transplant Network would love to sponsor his efforts. This is potentially really huge, especially for us in the hair transplant industry. Even if we were able to regenerate 50% of the donor hair, that would be enormous. I mean, that would be
Yeah.
it's one of the biggest breakthroughs in recent history. Uh, what's the next question?
All right. Next question by Peter Lustig. Is it possible to up the doses in the next test, or would that have any impact on hair count?
Is it
Yeah.
possible to up the doses?
I mean, again, we're gonna move ahead with the next trial, and I think we've had several options on doing that. But the most likely what's going to be happening is that it's going to be another run on an FUE. I know some colleagues are interested in FUT, so I don't want
Yeah.
to just waste our efforts collectively on doing 2 FUT studies, especially that verteporfin is not really yet easy to attain.
Mhm.
So whatever we have, we just want to make sure we're using it efficiently. So
Right.
I know some colleagues are doing the FUT. So everyone who's really looking forward for an FUT study, I think some colleagues are interested and are pursuing that hopefully soon. For me, what I would be inclined to do is another FUE study. Um, the details are not yet solidified, so we might do a bigger FUE, of course, or we might do bigger FUE areas. But I think what we're going to do is we're going to start with the 0.4 dose and up the dosage from there. So I believe the 0.4 dose has been most likely a good visual, the one with the most visual impact, to me at least.
Mhm.
So I would like to start from that and maybe use a higher dose of the same concentration and maybe a third with a higher concentration. So, of verteporfin itself.
Mhm. Okay. Yeah. I mean, I think playing around with the dosages is going to be key. Um, I,
Yeah, but the thing is, it's you're trying to get as much sort of I wouldn't say like, educated guesses with these
yeah.
doses. So and at the same time having, one patient at a time, and volunteers that are willing to follow up regularly. So this would be, we were trying to get, the best possible dosage out of each trial. And I think the first one has been, as apart from any limitations it has been a very good eye opener,
Mhm.
to explore further things and to improve on certain aspects in the subsequent studies.
Yeah, and I mean, I think thus far we have actual photographic evidence that there's a difference. So regardless of what anyone thinks, I mean, it actually works. At the very least, it's making a huge difference in terms of scarring. At the very
Yeah, I
least.
mean, you have the zoomed out photos, Melvin,
Go,
because I
go to the
know a
next
lot
one,
of
Jerry.
people
On
were looking
yeah,
at
hit
it.
next.
Um,
Yeah, right
no,
there. Then
this
we
is
have.
actually this is by the way, just to clarify, this is just a zoomed in, a magnified photo of the biopsy site. So, the C, the control one, this is the biopsy site of the control area, and the T is the
Oh, okay.
biopsy site. So the scarring here is not injected with verteporfin, or it's just a stitched scar after the removal of the suture,
Oh, okay. This.
or a few
So
days after. But I mean, this is just not an actual injected FUE punch. This is
okay.
after the
Go
biopsy.
there.
But
Oh,
there's
okay.
other ones. There are
Yeah,
other ones.
I don't. I don't have any other ones.
The
These are
ones
the
I've
only.
sent, there's ones that I've sent recently, today maybe, or with different dosage, and each.
Oh, yes, yes, yes. Um, I have them. What I can do right now on the fly, guys. Sorry, this
Yeah,
is live, but I may just have to airdrop it into. Let's see if it goes through. Airdrop it into the. All right, so we're gonna have to put this on the that same document. Uh, so yeah, so we'll put it back on the same Word doc that we use. So, but in the meantime, let's take
hmm
another question. In the meantime, that we do that. Well, we do this.
So the next question is by Niche Guard. In the next experiment, is there a way for us to mark the locations where hair follicles were taken off?
Okay.
Yeah, absolutely.
Yeah.
For the next trial, we're gonna pigment and tattoo the areas. So we're gonna use an SMP technique to do that. So at
That's
least
exactly the.
randomly tattoo. I mean, I wouldn't tattoo every single FUE, especially if it was a full FUE procedure. That would just be endless. But what we'll do is we'll just tattoo around, a significant number of extracted areas, and at least that way we can just easily go back to the scarring or the scarred punch sites.
Yeah, I was thinking actually exactly that. Do SMP so you can track it. You can see exactly where you harvested the graph,
hmm
and it'll be easy to track that way. I think
Yeah,
that's
absolutely. I
probably the
mean,
best
the
way.
next trial, it has been, obviously, we were waiting on the biopsies, and we were waiting on more or less a longer healing time, but also working on dosage, but also most importantly, trying to fix as much of the limitations of the initial trial as much as possible. So trying not to rush into anything. That doesn't mean it's not going to be moving forward, but it's just trying to get every single thing as much as possible thought of um,
Yeah.
before moving ahead. Because as I said, it's precious in terms of volunteers, and in terms of material, and in terms of the actual trial itself.
Yeah. I mean, I I don't think there'd be a shortage of volunteers. Uh,
I absolutely understand. The only thing is, realistically, volunteers would require, extensive follow-up. So this is
yeah.
something that, is not easy on anyone.
Yeah. Absolutely.
I mean, I'm sure if it was just a once-off FUE procedure, that would not be a problem. But
To
to
follow
be
up every
asking them
few
to
weeks.
follow up every
Yeah.
few weeks initially, and then monthly, it can be a burden.
Yeah. So let's pull up. We got it. We got it on here now. So let's pull it up. Is that the one? Yeah. Hit next. Make sure this is the one. Okay, perfect. All right, so these are more. So go back to the first one. I just want to make sure this was the right one. So this is a test site. If you want to walk us through this one, Dr. Barghouthi,
Yeah, so
this
this
is
is the
the.
point. I mean, I did not clearly clarify the square areas, but you can see
Mm,
it's actually a 2 by 2 centimeter square below each label. You can probably see the borders of each square roughly.
mm, yeah.
So this is the 0.24 milligram box. Um, Probably the least visual difference between both, but it's still, in my eyes, I see a slightly more density in the test area as well. But that again, is not enough evidence. If we move on to the 0.32 and the 0.4, maybe.
Yeah, let's go to the next one and see. Oh, no, no. Yeah, just hit next one. This one is.
So this is the 0.32. This is the one where we biopsied again. So if you can see, yeah, I mean, this is also a zoomed out picture because a lot of uh, people who are following and ask for as zoomed out as possible just to
Yeah.
see appreciate the scarring, the visual impact on the scarring itself. And also, yeah, so again, this is the same one that
The same one.
the
So let's
biopsied.
go to the
And
next
then the
one.
0.4. No, there's a
No.
0.4 milligram one.
Go next. Is that the 0.4?
No, it's labeled actually.
Um,
It's also in red and labeled 0.4.
um, oh, let me once again, guys. Sorry about that. Uh, that must have been the only one I literally missed. So we gotta do the thing over again. But in the meantime, there it is, 0.4. So we just gotta add it back into the document. Um, but in the meantime, let's get one question that Dr. Barghouthi could answer really quickly.
Okay, our next question by Dark Prin to 2500. What does Dr. Barghouthi plan to change slash modify in the next trial?
That's a good question. So, like, what are the things that you found in this trial that you're going to fine tune for the next one?
Yeah, so we're going to fine tune the dosing, which we mentioned. We're going to
Mm.
fine tune the sort of coming back to the same areas that we're studying. So we want to identify visually the sites that we've been looking at just to track the progress, by the dot basically. So we're gonna the two areas that we can follow up
Mm.
regularly. We're gonna modify the delivery of the medication. So we're gonna look at different injection techniques that are reliable or sort of more persistent or consistent. I mean, with delivering at a certain depth. So we may use a meso injection or a dermal stamp or something that would deliver
Mm.
at the same depth um, across the sites. Yeah, so and again, statistical stuff. So having the actual hair counts, maybe repetitively with follow-ups, getting the hair counts again, just to keep the data more consistent.
Yeah, getting like a target area hair count, right? To
Correct.
kind of make sure, and then you could do it in look at the control area, target area hair count where you did the extractions, and then pin the name area and the test site, and then we could actually statistically see uh, how many hairs grew back. We got it now, so let's go to the 0.4. So here we're
Yeah, I
looking at the
mean,
0.4.
yeah. So I mean, this is to me the most visually significant.
Yeah.
You can see on the right there that it's just taken at a different, not angle, but it's just sort of slanted slightly. But you can see that the actual scarring in the control area is a bit more. It's actually almost looks over harvested, although they're the same number of grafts removed.
Yeah, I mean this one looked to me, I was like, wow, this was a bit shocking to see how good the test area looks. compared to the controlled. I mean, it looks
Mm,
like you would assume that you took like triple the amount of grafts from the controlled site
yeah,
just by looking at it visually.
yeah. I mean, and again, if you zoom a bit further back, it's probably more difficult to appreciate. So this is as far out as
As you
possibly,
can.
reasonable to appreciate things.
Yeah. Yeah, this is huge. This is big news. Do we have any more questions, Jerry, that came in?
Yeah, we have one by Game One Ultra. Um, is there a scar revision planned where already existing scar tissue is cut out?
Good question. So I'm assuming he's thinking like someone who maybe had a previous FUT scar
Yeah, yeah.
and they want to get it revised and they want, I mean, I think verteporfin would be absolutely perfect for that. But what are your thoughts?
I mean, absolutely. Everything is possible, and there are several, probably 10 studies that could be done on verteporfin. Even I get emails about scarring, alopecia, and all of that, and they're all valid. It's all possible applications. What I would suggest, or what I would think, is to try and get a fresh FUT scar first. Um, See how the scarring is in a sort of a new FUT or a small FUT scar, before doing it on a revision, because we don't know what the underlying mechanism might be affected in a revision scar. Technically,
Mhm.
it should have the same effect if you remove fresh tissue and you resuture it and then inject verteporfin. It's like a new scar again, but probably if someone is gonna pursue with an FUT, I think they're gonna do it. They're gonna do it on a fresh one, or an previously injured tissue.
Yeah, yeah, that makes sense. Um,
But it is definitely valid that it could work well in scar revision.
yeah, not so much an FUE over harvesting, unfortunately, because
Yeah,
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 which that
because what
seems to be more prevalent nowadays.
yeah, I mean, unless you sort of microneedle maybe into the
Mm,
older scars and injure them anyway and then inject again, might
that
be.
yeah, that's a possibility. Let's get some more questions here. We have actually quite a few. We're trying to go through them here.
Okay, this one again by Niche Guard is how do we ensure that each area receives enough verteporfin?
Um, so how do you
Again,
ensure each?
the drug delivery mechanism is what's going to be a key in the next one, and it's just to give, the exact sort of dosage at the exact depth across the sites. So that's something that's going to be important.
Mhm. Yeah, that's a big one. Um, let's see, what else do we got? What other questions do we have here?
Okay, did the full FUE planned for the 18th of March happen?
No, that was the FUE. We wanted to do a full FUE in March. The only thing is, it was difficult to assess what dose to get and to do the whole procedure with one dose or to just split the area into two halves. Uh, I just felt that there might be the volunteer is there, he's ready, but I just want to
Mhm.
make sure that we utilize his efforts in as much efficient way as possible. So I didn't want to just do a non-verteporfin half donor and then another area with one dose or to rush it before
Yeah,
making sure that we have all the study limitations completed. But again, the volunteer is there, and it will be moving forward. But it's just we're trying to figure out certain limitations that we might to overcome.
okay. Um, Let's see. What's the other question? Looks like we have a
Uh,
long one here.
is there a way HRN or Hair Restoration Network can help get more doctors involved for us to have a global effort?
Yeah, I so the I my plan was to actually go this April to the Los Angeles Hair Restoration Conference with Dr. Umar, where a lot of doctors will be there participating and talking. And I plan to show them this video, show them this evidence, and hopefully pass their information over to Dr. Barghouthi so we can orchestrate efforts. I believe that Dr. Bloxam will be doing a verteporfin trial for FUT. That's the name I've heard. Is that the colleague you were talking about, Dr. Barghouthi?
Yes, but
Yeah.
I'm also in contact with other colleagues, and they're looking at different ways to help. Once they source or find a possible way of regularly sourcing verteporfin locally.
Yeah, absolutely. And the more doctors join in, the better. Dr. Farjo was another one that was interested in doing a study. I'm not sure if you ever spoke to him.
No, but hopefully in June they have the British Association meeting as well. So hopefully, if that goes ahead with that.
Yeah, and that would be I mean, he's at the forefront also of, he's got the hair clone company doing research, so he would be a big one if he joins
Hmm.
in and does the study for the UK. It would be pretty big, and he's got the UK actually backs him up on his research, so
Hmm.
that would be a good one. Um,
Definitely.
let's see, what other questions do we have?
All right, next question by Kaz. What improvements to the injection technique does Dr. Barghouthi have in mind for the next trial? And then follows up with, I remember something being mentioned regarding more even distribution.
Mhm.
Yeah, so it would be, ensuring delivery of the same amount and also uniform in depth of the injection site. So these would be the main things that we need to focus on.
Mm, perfect. Let's see. Do we have any other
Is there
questions?
any contact to Stanford?
That's a good
No,
question.
actually, no, no. Unfortunately, I mean, but no, it hasn't been. There hasn't been any contact with them. I'm following up on anything that they, would be publishing or any sort of original manuscripts that they would be publishing, because that could be very helpful as well, especially with any dose changes or any follow-ups. But no, there has been no direct contact yet.
Yeah, and again, I think just to say again, I think Dr. Farjo and his team at Hair Clone would be perfect because they have a lot of contacts and they work with universities, and I think they would be able to hopefully get a good contact with Stanford, and we can all, if we can all get this crowd funded and really make it, have the resources to really do multiple studies, I think that we're onto something huge here.
Yeah, I mean, definitely, it's something that we have to move ahead with it, but also move in the right direction and make sure everything is well planned. Just to at least provide an initial sort of pilot, trials that are you know of a good caliber.
Yeah, and I want to thank you, Dr. Barghouthi, for doing this trial. I think the hair transplant community owes you huge. The hair loss industry as a whole owes you huge. Just for those who are joining in late, if you guys want to contribute or want to help Dr. Barghouthi in his trials, go to verteporfin.org. This is a website that was created for him by users of the Hair Restoration Network to help him in his clinical trials because, of course, these things do require some resources to continue. Do we have any other questions here, Jerry?
Yeah, Kaz is asking, how is Arcturus donation going to be used?
So definitely, I mean, first of all, I thank everyone who's been helping in any sort of way possible, from the website, from any funding. So definitely, this is going to be something that is helping us with moving forward, at least with the next trial. So we're going to be focusing on the next trial. That's why it has been pulled back, because I think we could, moving with the funding, we can move with a better sort of equipment, study design, and all sorts of allocation of resources. So this has been very helpful in that we sort of looked back at replanning the whole next trial. But definitely it will be utilized in the next trial.
Yeah, and just so that just for the people who out there who don't know like how trials go, um, even to do a trial to in a university, it costs a ton of money. You know, the pathologist needs to be is not free. The resource, the centers are not free. So all of all of the funding would go into making the study better. A large
Yes,
you
definitely.
can fund a larger scale study. Right now, it's just one
Absolutely.
patient. Then it could be multiple patients. You could do it in a bigger facility. You could do it then in a hospital. So that's where the funding would go towards. Right now, it's very small because first it was coming out of Dr. Barghouthi's pocket. For those of you who don't know, he just did it on his own. But now with some funding, we can make it better and we can do it better, bigger. What's the next question?
Okay, by Jamie L. Could you drip the verteporfin into the wounds instead of injecting it?
Uh,
Mm,
yes, technically you can, and we thought of ways to actually use it as a dressing, or have it. I even thought about using it as part of the tumescent, but I'm actually worried that it might dilute it a lot,
mm,
especially with bigger cases. But you can probably soak it. The only thing is, I'm not sure how evenly distributed it would get if you just soak it in. And as well, you'd have a lot of it going into uninjured tissue or unextracted sites. So I would prefer to have a better sort of injection drug delivery mechanism that would ensure that it actually reaches in around the extracted areas more evenly, rather than just, because if FUE sites are just that there's normal skin between the FUE sites, the punched sites.
mm.
So I don't
True.
want the medication to just be sitting on the skin surface of the other areas, and
Non, yeah.
that would just be a waste of the medication. So we just want to make sure that it actually goes into the part where we want it to act on.
The harvested extraction point.
Yeah.
Yeah, that makes sense. Let's get, is there any other questions?
Yeah, next question by Juice God. If a verteporfin causes regrowth, is it plausible that simply wounding the recipient area and injecting verteporfin might cause hair to grow?
Absolutely, and that's another test or trial that is. I was actually in between doing that or doing an FUE, but I thought the FUE would probably, given that we have an experience with the initial trial on the FUE one, I think we should improve on that and move ahead with an FUE trial for the second test. But definitely, it might be something worth considering. It is doing sort of microneedling or injuring the recipient area and then injecting verteporfin. It is definitely a trial that I'll be interested in doing. I just think it should be maybe the next one after the next FUE.
Yeah, and there's just so many questions with that, right? Because
Yeah.
my question is, like in the Stanford study, the it regrew the hair that was already there, so that was already terminal. So my only concern is if, like, let's say someone is getting it wounds in an area that's void of hair, is it really going to regrow that hair? Is it necessary for you to
Correct.
have like miniaturized hair there, and then maybe it could
Mm-hmm.
regenerate it? I mean, there's just so many questions to that. I mean, that would open up a huge Pandora's box of different clinical trials and stuff like that. But it's exciting, definitely exciting.
Definitely. I mean, I know there's a skepticism about that, whether it's just going to regrow in a previously miniaturized area or not. But the reason I'm still interested in it is that somehow it's probably a simpler study to do.
Mm,
I'm not saying it's just at least we can have one square box maybe in a Norwood 6 patient, and we can inject in that and just monitor it.
yeah,
Um,
that's true.
but again, we have to have the scientific evidence, otherwise we should allocate the time and the efforts into something that we can probably see a difference with.
Yeah, yeah, definitely. I mean, I think, in my opinion, verteporfin is probably going to be best with the hair transplant, um, because, like I said, if you harvest 1,500 grafts and at least 700 grow back, almost half, that would be huge. Then you redo it again. I mean,
Mm-hmm.
your donor can go from 6,000 to 12,000 grafts, or
Correct.
for those people who
Correct.
have 10,000 grafts, it could go from 10,000 to 20,000 grafts. I mean, that's enormous. But I always think that it's going to have to be combined with hair transplant surgery, which I know is something that people don't want to hear, right? They want something to just, I wouldn't just get an injection and regrow my hair. Unfortunately, I'm not sure that's going to be the case, but who knows? We don't know.
Yeah.
It's
Yeah.
up in the
True.
air. Let's see. Let's get another question.
All right. Will the FUE patient be different from the first trial? And do you have a timeline? When do you think you should be able to start the next trial?
Good question.
Yeah. So basically, the FUE patient is a different patient. Yes, it's not the same volunteer. So that would be a different one. He was originally booked for the 18th of March, if I recall correctly, but then we just wanted to make sure we get few changes done. So I have him booked I have him booked for 2 slots. One of them is in towards the mid end of April, and if not, it's gonna be just early May. So it's gonna be fairly soon. That's for sure. I just need to have 2 dates for him, just to suit his timetable as well.
Okay, perfect. Yeah, it's good that it's going to be a different patient too, because I feel like
Yeah,
that
yeah, definitely.
that that's going to make a big difference. The more people, the better. Um,
Yeah.
let's see. Let's get another question here.
Let's see. Okay. Kaz is asking, in a really long FUE session, say 8 hours, would you wait until the end of the surgery to inject a verteporfin?
Yeah. So first of all, the extraction part, which we were concerned about, is not going to be 8 hours. So it's going to be around 2, maybe 3 hours. Um, so what we'll do is we'll do all the extractions, we'll do the injections, and then because we want to cover it and protect it from the operative lights and the actual lighting, because verteporfin can be photosensitive. So what we're gonna do is once we inject it, we'll cover up the area and we'll keep it covered, and then we can carry on with the recipient area.
Nice. Yeah.
At the same time, even if we delay the injection towards, the end, even after the implantation, that shouldn't be a huge delay, because as soon as we have a window of time before the scarring mechanism starts, so that shouldn't be an issue. It's still t equals 0, when you inject on the same operative day.
Mm.
So you're still operating. You're not. There has been studies where they injected few days after, and they weren't as conclusive. But if we inject on the same day, I would tend to probably inject it once we finish all the extractions.
Yeah. So I mean, you're saying so even if the extractions take, which you said it usually takes around what, 3 hours. Let's say a 3,500 graft case would normally take around what, like 3 hours, maybe 4 hours of the day.
Uh, Probably, yeah. I don't do 3,500 in a day anyway, but it takes around 2-3 hours for like maybe 2,000, 3,000.
God, yeah. So I mean, after a few hours, it's still not going to make a difference, is what you're saying.
Yeah, as long as we inject and then we cover it, that has to happen, and then we can move ahead with the rest of the procedure.
Right. Let's get a next question.
All right. Do you think we can combine the FUE trial and recipient injection in one trial to save time?
It's a good question.
We can. The only thing is, with the recipient patient, we want a clear area of no hair, or we want to be able to monitor the area. The FUA patient is going to have his grafts transplanted, so that's going to complicate things in terms of follow-up. Where would we measure? We can't leave him an empty area where we did not transplant just to inject verteporfin in it. So that's one obstacle, or that's one technical difficulty. The other one is that we're gonna be, giving him an FUE dose of verteporfin and also one in the recipient area. So maybe dosing wise, it's gonna be harder. So I think it's always better to split the patients,
Yeah.
and study them individually. There might be, I think doing the recipient area shouldn't take long, even after finishing the FUE one. So we'll probably move on with the recipient trial shortly after that.
Mm. And I mean, when you were saying that, my first initial thought was, what if you did an FUE to the hairline, and let's say like a Norwood 6, and you did verteporfin in the crown. So you would transplant all the grafts to the hairline, but you have the crown that's wide open, that has no hair. But
It's true, but then when you do a trial, you have to. That's what I'm saying, is that each trial teaches you a lot of things for the next one. So, for example, you need to cover the verteporfin injected area. So then you would have, the donor area covered,
the crown
the crown covered, and then the crown is close to an FUE
site.
transplanted area, which you don't want covered. So, in terms of, I don't want anything to go, mess up with the patient, even if nothing works, at
Yeah,
least you want his hair transplant to work.
I hear what
So,
you're saying. Yeah. The more things you throw
I
into a
don't want,
trial, the more complicated you make it, the more chances of it getting
again,
skewed.
for example, we don't want it wet for verteporfin, and then at the same time for the transplant, we want to make sure that he gets his wash the next day. So it's
Mm,
gonna be, um, challenging to add different variables at the same patient.
yeah, that makes sense. Um, Look, do we have any more questions?
We do. Did the patients report any side effects?
That's a good question.
No, no, no,
That's a really good question. Yeah,
no,
that
no.
was yeah. No side
And to be honest, I know I've seen a bit of hyperpigmentation in the first few weeks,
effects.
and maybe even lasting the first couple of months. But when I saw him recently, there was nothing different than any normal skin. in terms of discoloration or hyperpigmentation.
All right, perfect. Do we have any other questions? I mean, I'm sure we have. We keep getting them.
Let's see. Next question. Do you think there needs to be unscarred skin surrounding the treated wound?
Do you think there needs to be unscarred skin?
I guess maybe a.
Not necessarily. What I think needs to be is that you should have a scarred wound to like a fresh wound to inject verteporfin. So it has to be a fresh scar or a fresh wound.
I think what he might have also meant is does the scalp need to be virgin? Meaning, can someone who had a previous FUE and has already scarring
Yeah,
there, would it
I
still
don't know.
work?
To be honest, I wouldn't know. I don't imagine. I think verteporfin would work on the actual current
The new harvesting.
event.
Yeah.
So if your new harvesting helps with maintaining some of the hair or even improve the scar quality in that setting, then that would be the goal. But I don't think it's gonna be limited by uh, having other older scars.
Mhm. Yeah. Yeah. I mean, that was the first thing that I was thinking. I was like, well, what if someone like me who has maybe like 500 to 800 scalp grafts left? And would they be able to, let's say in the future, if this actually becomes something that there's enough studies and we verifiably see that this is working, would it be possible for someone like me to then get 500 grafts and then maybe that out of that 500, 300 grow back and then keep going until I can get that last bit of hair without completely depleting my donor?
Yeah, I mean, I don't know if you're gonna go for more surgery or not. I don't think you need it, but if you keep your 500, if you keep the last bit there, then that's very good.
Yeah.
If you can keep a certain low threshold of the bare minimum
Yeah,
consistently, then that's even an accomplishment.
absolutely. Let's see. Let's get another question in here.
Okay. Is verteporfin phototoxic or just photosensitive? For example,
Photosensitive
deac-
is what we're
deactivate
concerned
all
about.
light.
Yeah.
Photosensitive.
Yeah, I mean, so if you look at it, it's photosensitive. So it what in the skin, what we know is that it's gonna be inactive if it could be subjected to certain light.
Mm.
Again, we there's no such there's no evidence on the skin itself, but out of precaution, we decided to cover it. Again, for example, in eye surgery, it's actually activated by a certain laser. That's how it works in the retina. So it's a different setup.
Mm. Got it. Would it be? I mean, this again. I'm not just throwing it out there. What if like you combined it with like some sort of like low level laser therapy or something like that? What you think it would make it more effective, or would it? I mean, I don't know. I guess that's something to possibly test.
Yes, correct. Absolutely. I mean, I think there's one of the original patents on it was used along with low-level light therapy or some sort of light therapy when they were looking at the neovascularization in the eye and the retina. So yeah, I mean, the relationship with lighting or what sort of wavelength is something that we can look at. Um, but in terms of the skin, I think we would be probably in a safer place trying to avoid having the light deactivated.
Mm. Got it. Let's go to the next question.
Should we inject verteporfin before extraction as well?
Yes, you could, and I thought probably one of the ways to do it is to incorporate it into the tumescent fluid, but then I couldn't figure out the dilution impact on that. So, with tumescent, it's bigger volumes, and verteporfin is a very small dose, low volume sort of preparation. So, to dilute that further, we might not get the impact that we're looking at. But yes, we can certainly inject it before. The only thing is, again, we're going to be injecting a bigger area, and at the same time, we're going to be extracting a quarter of that area,
Yeah.
eventually. So some of the medication is just going to be there in the entire area, and it's not only the volumes, but the volumes we don't want to exceed any sort of toxic levels.
Yeah, and you also don't want to waste any of the medication, right? Because it's not easy to source.
Yeah, but I mean, but again, we don't know the volumes. We don't want to overdo it as well. So
Yeah.
if you're gonna inject the entire donor area at, let's say, an interval of 0.5 or 1 centimeter, then you're gonna do a lot of injections. At the same time, you're not gonna be
Pinpointing it.
extracting all of these areas.
Yeah,
Anyway.
that's true. Let's see. We got another question here.
Okay. What are the doses that you think you will try the next trial? Will you have more test sections, more dose concentrations than the first trial, or will it be the same?
So we're inclined to start with the 0.4 as the lowest dose and go up by 1 or 2 doses. And then I believe we're going to be doing one with a higher concentration of verteporfin as well. So we're trying to get the 3 things mentioned together. But again, it depends on the area, and we need to have a control area as well. So depending on the size, we might have to just go at least with the higher dosages first and then change the concentration maybe on a subsequent smaller study.
Yeah, it would be, and I think it's also going to be harder to, if you have more extraction sites, like instead of having one block and one controlled site, having 2 of those, I don't know if it's as easy to be as accurate. I think,
Yeah.
having just
Yeah.
those 2 is going to be, you're going to be able to
So
be
I
more.
mean, we know the current concentration is working somehow.
Yeah.
It has been safe, and at the same time visually, pathologically, promising, hopefully. So I think we might just try to increase the dose slightly on the one of the best dosage that I've seen, which is the 0.4. So we can increase that maybe by 1 or 2 intervals before changing the concentration.
Yeah. Let's go to the next question.
Our next question, is it worth testing Verteporfin also on the recipient side in a transplant to see if it helps with the number of grafts that take? I believe it's 5% of grafts that don't survive.
Yeah, I mean that's an interesting one. So I think he means like, when you put you're obviously creating recipient sites, so you're creating some wounds, then you're placing the grafts into those recipient sites. I wonder if you inject those sites with verteporfin, if it would actually help the growth, if it would help the yield. That's an interesting one.
Definitely interesting. The only thing is, again, if let's say the survival difference is not big between both, then we just need to find a way to, statistically measure that, the significance of verteporfin in the improving the growth rate or the survival rate. So again, it'll be 2 different areas, one with verteporfin and one without. I would I was thinking of it as maybe when that happens is using it as a holding solution. So you can actually have the grafts in a solution of the saline with verteporfin, and then once you have the recipient sites made, you can just implant them, and they'll be soaked in verteporfin basically for the last while. So maybe that's something that can be
Yeah,
considered as a holding
as a
solution.
holdings. Yeah, no, that makes sense.
But it's difficult again to quantify the improvement, because some hair, good hair transplants, they would, as I said, maybe the survival or the non-survival rate is low. So how would we quantify the improvement that verteporfin is causing?
Yeah.
Is just going to be, sort of limited.
I think the one way is like Dr. Shapiro's study on FUE versus FUT, where he had one side was
Yeah,
FUE, the other side was FUT, and so you could do one side, injected with verteporfin, the other side just regular, no verteporfin, and kind of measure it that way. But
yeah,
I mean, the holding solution makes a little bit more sense in it being just more efficient with the hair transplant itself, right? Because if you already have the grafts there soaking in the verteporfin, then you place it into the sites, you're already having to place the grafts in a holding solution in
yeah.
most cases. So nothing really changes in your process, and you could just carry it out just like you normally would any hair transplant without having to, individually inject these recipient sites, which would be more labor intensive.
Yeah, I mean, the only thing would be the photosensitivity aspect of it,
Mm,
because we'd be transplanting with lights on, and
that's
and
true.
it's yeah, each trial is, it's maybe simple to come up with thought of it, but then the actual steps makes things sometimes harder to implement. That's the whole
Y-
problem sometimes, with trying to figure out each step. And if you don't, then you're kind of wasting a precious step.
Yeah, yeah. It's like the saying, easier said than done. Right. That's yeah,
Yeah, because you get up, you plan everything, and then you find that, okay, you're eventually you're doing a clinical procedure as well, and you need results with it, because these are volunteers who are not only volunteers, but some of them are actual patients, and they want a hair transplant. So if you're using verteporfin in that sense, and then you're following the standard procedure,
mhm,
you might have no effect of the verteporfin, for example, with light or with
yeah,
modifying the steps.
yeah. No, absolutely. Let's go to the next question.
Okay, if a patient comes to you wanting to sidestep the scientific method and just have their entire donor area treated, would you do it?
I don't really get. So would it be
So he's
as in sidestep the scientific method?
yeah. So instead of like doing any trial or anything like that, like he's just, I don't care, let's just do the whole 2000 grafts, just do the whole thing verteporfin, and let's just see what happens.
Yeah. So two things. I mean, we could be just a bit far from the effective dose, and that would just mean that he did not get the best outcome possible. Um, you know, let's say if we use one dose for him, and then we find out that maybe a higher dose is even better, then that's something that he missed on. But more importantly is the safety aspect. So if someone just wants to have the entire donor injected with the verteporfin, we need to make sure that it's safe enough to have that volume in it.
Yeah. No. And I
But what? Yeah, I mean, but definitely the ultimate goal would be is just to have it as part of the procedure, you know. So once we figure out good dosing, good safety, volumes, all that, you know, this is something that's gonna be hopefully incorporated in a standard procedure.
Yeah, I mean, we're on to really groundbreaking stuff, guys. I mean, I don't know if it really is sunken in. Um, but I for the people who are just getting hair transplants now, or the people who will get hair transplants in the next 10 years, my gosh, it could be just completely different than how it is, how it was just a few years back, you know. Let's go to the next question.
All right. What about one trial where hair is transplanted to the frontal third and mid scalp and leave the crown for testing involving area injections?
Oh, yeah, that's what I had proposed earlier.
Correct.
Um,
Yeah. So yes, I mean possible, but again, different protocols to follow. The transplanted area cannot be treated in the same way that the injected area is going to be treated for follow-ups or even aftercare or immediate aftercare. So this is going to be the challenge with having different studies on the same patient.
yeah.
It's just different outcomes.
Yeah, it's always better to do it on separately, and it makes it the test more accurate. Let's get the next question.
All right. Do you think it only works on full thickness wounds, or might it work in combination with partial thickness wounds into existing scars with laser or micro needling?
Trying to understand that question. So, I think maybe he means like, will it work if you're not completely harvesting the graft? That's maybe what I'm getting at. Maybe he's saying like
Um,
partially transecting the graft and injecting it
I think it's actually addressing, scars in general, maybe not necessarily with,
with the surgery.
to do with hair, but maybe some partial thickness grafts or wounds, or wounds that have been treated with lasering. So
Oh,
um,
yeah.
technically, it's to do with the scarring mechanism. So anything that is scarred or is going into a scarring mechanism, verteporfin theoretically can, to some extent, inhibit that process. So I don't think there's any issue with the severity of the scarring or the partial versus full thickness wound differences.
What do you think about and this is was like a long time ago, there was a doctor, I forgot what his name was, Dr. Go or something like that, where he was claiming hair regeneration, but he was using a really small punch and just transecting the graft. So a three-haired graft, he would take one hair follicle and then two would grow
Yeah,
back. I'm wondering
yeah.
if verteporfin would be good in testing that, where if you partially transect the graft, will that transection grow back fully? Will that, instead of two hair root growing back, would a three hair root grow back? I don't know if that's something maybe in the future to look at as well.
Yeah, I mean, definitely. Uh, I don't know with partial resection or transections. I'm not I wasn't in favor of that
Mm.
technique anyway, in terms of, partial transections. Uh, I don't know if that's possible, to be honest. If partially transecting it would regrow the follicle itself, because um, we're looking at the actual scarring inhibition, and that would lead to the hair regeneration. So I'm not sure if you're actually removing part of the hair, then you would regrow it back,
Mm. Okay, yeah.
once it's removed.
Yeah.
It might remove back the one that is left behind, you mean, correct?
Yeah.
Is that what you're...
Yeah.
Okay, yeah. I mean, it is possible. but you'd have to just make sure you statistically document everything, and, make sure that what you left behind is just one hair follicle, not the 2 out of 4, or
Yeah, it would be a little bit more difficult too, because you have to. It would be a hard test, but it's just a thought that
because
came to my mind.
often you would see maybe a 2 hair sticking out of the skin, but then when you punch it out, it's 4 hairs,
Yeah,
a 4 hair follicle. So you wouldn't really know how much you're leaving behind, I think.
yeah. Let's get the next question.
All right, I think this is the last question up. So it says by S. Miller.
Okay.
So should I hold off on getting a hair transplant?
That's the $1 million question. I think that's the $1 million
Yeah,
question.
yeah, absolutely. I don't mean absolutely hold back, but I don't know. Maybe go conservative.
Yeah. Yeah, I mean, gosh, if I was researching this, if this was 10 years ago when I was looking at hair transplants, uh gosh, what would I do? I mean, it's exciting. It's like you don't want to be in my position, right? Where I've had a whole bunch and then, I'm kind of like not a good candidate anymore. But you want to be in a position where, you're right at the cusp of it. It's like it's rolled out. Um dang, what would I think that, and I'm going to speak as a patient. I think that if you're in a position where you're like a Norwood 2 or Norwood 3 and you're on finasteride or dutasteride and minoxidil and that's working for you, I would probably hold off maybe another 2 or 3 years. Let's see how things progress. And hopefully, once you actually need a hair transplant a little bit more, then this will be readily available and there's more studies and then you'll be in a better position. That would be my choice. But if you're like a Norwood 5 or 6 or something, and I would say probably no need to wait because you probably want to get a hair transplant now. And like Dr. Barghouthi said, be conservative and maybe try to just do the hairline and the mid scalp and leave the crown and have a good uh, donor reservation there. So in the event that you do want to try this, that there'll be enough hair there for you to get. Because if you get in a position like me, then it's like, you have the hair that's left over is not the best hair quality. There's not a lot of it. There's a whole ton of scarring. So, it's definitely not optimal.
Yeah, at the same time, if you have few surgeries done and you have good outcomes, even if you have 25-30% extra donor reserve left because of that, then you'll definitely reach your goals. You know, even most people would reach maybe 80-90% of their desired goal with multiple surgeries. So add to that, availability of maybe 25-30% extra donor, and then that's good enough as well to give you more satisfaction. So across, even if you have one procedure, you might be benefiting from the outcomes for the second or subsequent procedures if it's an advanced case. I wouldn't know what the exact case is, but even for advanced cases, if they start with a conservative first session, maybe things will be showing up later.
Yeah, definitely. I think are we past an hour? I just saw 2 more questions in, but let's make this the last one. I promise you, Dr. Berger. I know we're a little
No, not
past
at all. Not at all.
time. What's the question?
The last one by Jamie. Could you talk about sourcing this? Uh, is it because you are in Jordan that it's harder to get?
No, actually, I was talking to some of the doctors in Europe and in the US, and it's equally difficult. It's just The clinical grade one is out of stock, which is Visidyne. The research grade ones, which are, you know, a good research grade one, they're you know, you have to go through different labs, different ordering forms, and all sorts of stuff. So, you know, luckily I've managed to source some, but I wouldn't know whether I have to switch labs for subsequent ones, or you know, they can continue on supplying that. So that's the only thing.
Yeah, yeah. I mean,
It's hard to get because it's not of clinical use per se, you know, because with eye surgery or retinal disease, it's no longer used. So there are other medications that took over, but most companies use it for research purposes, and that's the one we're using. Um, and that's why you go through different channels ordering it.
I wonder if Visidyne, because that's the one that got FDA approved, if they would ever want to reapply for a new FDA approval for hair like to be used like clinically for hair transplants, or just, I mean, this could be used for anything, for any cosmetic procedure, any surgery, any surgical wound could technically be treated with
Yeah, I
Visidyne.
mean, if you know, I mean, if there is anything, you know, promising to it, I would see no reason why they won't, you know, go with adding an FDA license to it, or you know, another clinical use.
Yeah, yeah, absolutely. All right, Dr. Barghouthi. Well, I think we got a whole bunch of questions in there. Just before you go, and for everyone watching here, if you're interested in learning a bit more, go to the Hair Restoration Network. We have a pinned topic, Verteporfin trials, where Dr. Barghouthi updates us on the latest news. If you want to also contribute and help the clinical trials continue in the future, go to verteporfin.org, which is a website that was just created for Dr. Barghouthi. He didn't even create it himself, but verteporfin.org if you want to contribute as well. And everything will be linked in the description box once I finish the stream. So thank you again, Dr. Barghouthi, for all of your
Thank you. And
time.
just one last thing.
Yeah.
Just one last thing. So, you know, I have a lot of inquiries and emails about patients or people asking about different applications of verteporfin. I hope we've answered some of these here, but I promise I'll reply to the emails, even if if it's taking time, but you know, I will reply to these emails. And to the threads on Hair Restoration Network or elsewhere. And please, I mean, I know some of the patients or some of the followers, they are across different channels. So in some cases, it's just easier for me to answer in one place. So please feel free to share, you know, across the other groups. I know some or most people are across different channels. So
Yes.
just feel free to update the others, because it's sometimes difficult just to write the same message across different channels.
Yeah, absolutely. If you guys, I know you guys, there's a bunch of different places that you guys can go post, different forums, um, different crowdsourcing websites, or just anything related to hair loss or surgery. Share this, because I think it's important, and other surgeons may be interested, and they may not know, other patients may not know of this. So hopefully you guys can spread this video far and wide. I'm also going to, as a promise to you guys, the questions that I receive in the comment section, I will personally ask Dr. Barghouthi, and when he replies to me, I will put it in the comment section. So if you guys have questions that weren't answered after the stream is done, ask it in the comment section, and I'll communicate with Dr. Barghouthi to get it answered. Just before you go, Dr. Barghouthi, what is your email for those who want to email you directly?
It's info@vertexhair.com or taleb@vertexhair.com. The ones you have on the network. So
Yeah.
both of these, they're monitored. I just need to go through the emails personally at times. So for appointments, for things like that, the office can handle that. But for certain verteporfin questions, I'll have to answer myself. So it may take a bit longer, but I'll answer everything.
Yeah, absolutely. And guys, use me as a resource too. If you have questions, ask me, and I'll ask Dr. Barghouthi, and when he can reply to me, as soon as he can, I'll respond. All right, guys. Well, thank you so much. Thank you so much, Dr. Barghouthi.
You're
This
welcome.
was an
Thank you
amazing
very much.
conversation, and hopefully we'll do another one when you start your new trial.
Absolutely.
We'll do another live stream and discuss it and kind of get the input from the community. But thank you guys so much.
Absolutely.
Till
Thank
next
you.
time.